Hello and welcome back to Survey of Biology as we continue with Unit 2 on cell division. And if you remember in Section 1 we talked about the cell cycle. In Section 2 today we're going to continue on to mitosis and look at how the cell actually divides and splits into two identical daughter cells. So just a quick review, here's the cell cycle up here. So if you remember last class we talked about interphase, which was basically from the end of cytokinesis all the way through till mitosis happens again. So what we're going to focus on is what happens in mitosis, what are the actual phases there. And if you break it down into the phases, you can see that within the M phase we have 6 steps. There's prophase, prometaphase, metaphase, anaphase, telophase, and then cytokinesis is where you actually get the pinching off and we get 2 new daughter cells that are identical genetically. And that's what we're going to focus on as we go through these today, is what happens what happens in each of those phases to get us to our new daughter cells. The first thing to remember is that interphase is the vast majority of the cell. So a cell spends about 90% of the cell cycle in interphase. So that leaves only about 10% for mitosis and cytokinesis. And the difference between those two, mitosis technically is the division of the nucleus, and cytokinesis is the division of cytoplasm. So in conjunction, those two things cause the splitting of the cells into the new daughter cells. So as we saw before in the previous slide, the stages of mitosis are prophase, prometaphase, metaphase, anaphase, and telophase. So now let's go through and look at these individually. So here is kind of a color-enhanced picture of an actual cell dividing. So here's interphase up in the upper left-hand corner. And from here we can see the nucleus is in the blue right here, and the spindle fibers that are going to be forming are the yellow, and kind of the cytoplasm is in the red around. So what we can see in prophase, and it's a little bit harder to see, but you might be able to see 2 small dots here. So those are the centrosomes, the centrioles that will go towards the poles to pull it apart. And if you look in the nucleus, we still have chromatin. The DNA hasn't condensed. Now, as we transition to prometaphase in the upper right-hand corner, the nuclear envelope has dissolved. The chromatin has condensed into chromosomes, so we see these snake-like chromosomes, and there are 2 distinct centriole regions that will serve to pull the DNA apart. And again, the green in these pictures are the spindle fibers that are going to attach to the sister chromatids and pull them apart. In the lower left now we have metaphase, and here we can see that we've got the chromosomes lined up on the metaphase plate in the middle. We've got the spindle fibers attached to the sister chromatids ready to pull them apart. And in anaphase, that's when the spindle fibers will shorten, the chromosomes are pulled apart, and we start to get 2 new cells. And finally in telophase, as we can see here, we can see where the 2 new cells will be forming to give us our daughter cells. So now step by step as we go through, prophase is where the chromatin condenses, the nucleoli disappears in the nucleus, the nuclear envelope starts to disintegrate, and the centrosomes move away from each other. These are where the spindle fibers, the ropes, if you will, that attach to the chromosomes to pull them apart come from. Prometaphase is when the kinetochores develop. So these are areas of protein in the centromere region of the sister chromatids. So the kinetochores develop, the chromosomes attach to spindle fibers, and a tug-of-war happens so that they reach alignment in the middle. And when they reach the middle, that's what triggers metaphase. So here the chromosomes are in alignment at the equator of the cell. They're in a line called the metaphase plate, and something called APC, or anaphase promoting compound, is going to be released, which triggers the cell to start to pull the chromosomes apart and transition into anaphase. So anaphase is important because it's here that the sister chromatids separate. So we talked about how in the S phase of interphase that the chromosomes will replicate, and we get two copies held together at the centromere called the sister chromatid. It's at anaphase that those sister chromatids are actually pulled apart and they become individual chromosomes in the newly formed cells. So the centrosomes move farther apart, and the non-kinetochore fibers elongate the cell gets stretched out, and there's something called motor proteins that we're not going to worry about right now that help that movement. But as the anaphase continues and the chromosomes are pulled towards opposite poles of the newly formed cells, telophase commences. Here the spindles disappear, the nuclear envelope reforms, the chromosomes uncondense, they go back to chromatin, and the cell starts to pinch off, which leads to cytokinesis. So here's where we get a cleavage furrow forming. And imagine having a belt around your waist that's being pulled tighter and tighter. And that's exactly what happens. So there's this band of protein called actin that forms this contractile ring in the middle of the cells, and it pinches the cells apart until they finally split, and we end up with two new cells that have been pinched apart, and they're genetically identical. They would then go into G1 phase where they would grow, replicate their organelles, and get ready to function. So again, on the upper is kind of the electron microscope versions, and the below is the cartoon version. So as you can see, in G2, we've got the chromatin, the cell's getting ready to divide, and it's not till prophase that we see the chromosomes start to condense. So here's where we have our sister chromatids. The centrosomes start to form spindle fibers, and they migrate to the poles. During prometaphase, the spindle fibers will attach to the kinetochore region of those sister chromatids, and the tug-of-war will ensue where the spindle fibers are pulling them back and forth until we get to metaphase, which is where they line up on the equator of the cell. And at this point now, they are triggered. The sister chromatids will be pulled apart as we go into anaphase, and the individual chromosomes are now pulled to opposite ends of that cell. Telophase and cytokinesis leads to the cleavage furrow and the pinching off, and we get two new genetically identical daughter cells. So one thing that I want to spend just a minute on is what's happening on those spindle fibers when they attach. So the fibers are made of this protein called tubulin. We've talked about how they're assembled in the centrosomes, but what happens is the protein in the DNA at the centromere region, so right over in here on those sister chromatids, the spindle fibers will attach to those. So each of the chromatids kinetochore faces outwards towards the pole, and that's what allows the little tug-of-war to happen as they struggle to line up during metaphase. And so if we look at this picture right here, you can see in the yellow, there's your sister chromatids. We've got our centromere region right here in the middle, and part of that centromere are these proteins called the kinetochore, right along here. And it's there that these microtubules attach to. And that's what's going to help the cell pull those sister chromatids apart so that each new cell ends up with a complete complement of chromosomes that it needs to live and function. So thank you for your time, and we will see you next time when we start talking about meiosis.